Hello, my name is Namwa Winfred, and I'll be explaining about the exploratory data analysis for tomato leaf disease early detection. First and foremost, exploratory data analysis is an analysis technique that is used to identify general patterns within the data. It's the initial stage of data analysis, and it's used to pre-process data after data collection. The steps of the exploratory data analysis are data collection to gain domain knowledge, confirming data types and their probabilities, measures of central tendency, measures of dispersion, and others. The aims of the exploratory data analysis is to identify potential data quality issues, such as missing data, outliers, and inconsistencies. Its major objective is to gain insightful findings from the tomato leaf data set to inform subsequent steps within the machine learning model development. Key data preparation steps include data collection. Here we searched like different sites to find appropriate data for the tomato leaf project. Then data discovery and profiling, like you discover the data and then you rate about it, what you've gotten about it. Data cleaning to remove unnecessary data, to correct where necessary, and to maybe fill in where there is no data. Data structuring, data transformation and enrichment, like you transform the data into a form that is appropriate for the project. Data validation and publishing, to make sure, like data validation is making sure that the data is accurate and correct, then you publish it.
Yes, my name is Henry Senyonjo, and I'll start from the overview of the data set. It's named Tomato Disease Multiple Sources, and it contains over 20,000 images of tomato leaves with different diseases. It has 10 classes of diseases and one class of healthy ones. And then it has images, and the images were collected from both lab scenes and in the wild scenes. The goal is to develop a lightweight model that can predict tomato leaf disease and deploy it offline on a mobile app. That is, we shall create a mobile app where our model will be used. And the link here, this link is the source where we got our data set. Then the reason as to why we chose this data set is because it contains a diverse range of images showcasing different stages and severities of diseases affecting tomato plants. And then it also contains a large number of images. That is, each class has very many, or a large number of images for each disease. And then the dataset also has high quality images. Then this is the sample of the images from the dataset. The images have labels, as you can see. We have one from bacterial spot, and then the olive blight. Then we have the healthy ones, and then all the other classes of diseases are all represented in this slide. And then we have the training and the testing data, which is stored in subfolders and then subfolders, where we also have the image count of each class of disease. Yes.
My name is Ria Drake, and I'll start from where my colleagues have stopped. So I'll take you through the code section, that is the codes we wrote to do exploratory data analysis. So we use Jupyter Notebook and we use Python to do the exploratory data analysis. So first, we wrote this code to show one image from every subfolder inside the main subfolders of this data set, because it has two subfolders, that is the that is the trained data set, and then the valid data set used for validation. So first, we have to install OpenCV, which is just to open images and like folders. Then we imported OS. Well, OpenCV installed it, then imported OS, imported CV2, matplotlib, that is used for drawing these bar graphs and histograms. So first, we accessed the main subfolder, which is stored on this PC, which is named dataset. Then we had to transverse into like the subfolders, that is train and valid. Then into those subfolders, there are different subfolders of different tomato diseases. So we display these images, that is bacterial spot, alebrite, and all those health ones and all those. So to know like whether we are really having the required images. Then next, wanted to find out like different number of images in every subfolder. So I had to traverse through like all the subfolders again, and then counting every image inside every subfolder. So we found out that we're having train subfolder and valid subfolder having more subfolders, that is bacterial spot having 1043 images, alebrite having 281 images, and all others, as you can see. Then wanted to plot a graph that's showing like how these images are being how images are being stored in every subfolder. So we have bacterial spot having like 3,500, then health spots, red bright. So that is the graph. Then we used this scatter plot image. It is we first imported matplotlib, which is used for displaying this scatter plot image resolution. So that is the quality. So most of the images are between 1,000, that is by height, and then 1,000 by width. Then we want on like to display the color distribution in every image. So we used bacterial spot as an example to show like number of like how this color is displayed, that is green and red, as you can see. So if it is affected by bacterial spot, red is higher than the green. That's why we are seeing only red here. Then for others, there can be a combination like average display between red and green and all that. And then we stopped by displaying like the color correlation analysis of the whole data set, as you can see.